Yes, I hope everyone got the link. Please check your chat box. So there I had mentioned the link. So when you click on it, you will get the biology notes, which we recently finished in the live sessions. Right after many days, I got a chance to meet you all and to discuss the biology concepts, right? So in the last session, ಹಿಂದಿನ session ನಲ್ಲಿ, we discussed regarding the principle of inheritance. Multiple choice questions, the principle of inheritance. And we are aware that maximum marks in our question paper will be comprising the questions from this chapter. So around 14 marks, right? Around, 12 to 14 marks we will get from this chapter. Okay. So today we are going to discuss regarding the short answer question, that is 2 or 3 marks question from this chapter, as well as few 5 marks question. We need a lot of time for 5 marks question because 5 marks question is compulsory from this chapter. So 1 or 2 questions you will get from this chapter under the 5 marks domain, right? So let us discuss. Yeah, please, I request again everyone to check your chat box and let me know whether you got the link. How to check the chat box? Just click on the mobile, click on 3 dots. Yeah, yes, Sita, okay. Click on 3 dots and click on the chat box. There you will get the link. Okay. Just a minute. Okay, fine. So, Deepak, just check the chat box. Just a minute. Check it now. You got it. Let us begin. So, principle of inheritance. From the blueprint, that is from the previous year question paper and 2 or 3 year question paper, when I analyzed, I got to know that. So we will get multiple choice question 2, 2 marks question 1, long answer question 2. So I'm telling you, so we have to focus more on long answer questions, right? So we already completed with multiple choice questions. Let us focus on 2 marks and 5 marks question. Right, fine. Deepak, I hope you got it. Okay. The first question, differentiate between linkage and recombination. Differentiate between linkage and recombination. So what do you mean by linkage? So we know that a scientist named T.H. Morgan ಅವರು ಏನ್ ಹೇಳಿದ್ರು, ಲಿಂಕೇಜ್ ಬಗ್ಗೆ ತಮ್ಮ ಡಿಸ್ಕವರಿಯನ್ನ ಪ್ರತಿಪಾದಿಸಿದ್ರು. ವಾಟ್ ಹಿ ಟೋಲ್ಡ್, ಸೊ ದ ಜೀನ್ಸ್ ವಿಚ್ ಆರ್ ಪ್ರೆಸೆಂಟ್ ಆನ್ ದ ಸೇಮ್ ಕ್ರೋಮೋಸೋಮ್, ಯಾವ ಜೀನ್ಸ್ಗಳು ಒಂದೇ ಕ್ಯಾರೆಕ್ಟರ್ನ ಎಕ್ಸಿಬಿಟ್ ಮಾಡುವಂತಹ ಜೀನ್ಸ್ಗಳು ಒಂದೇ ಕ್ರೋಮೋಸೋಮ್ ಮೇಲೆ ಇದ್ರೆ, ದಟ್ ಫಿಸಿಕಲ್ ಅಸೋಸಿಯೇಷನ್ ಅದಕ್ಕೆ ಫಿಸಿಕಲ್ ಅಸೋಸಿಯೇಷನ್, ಫಿಸಿಕಲ್ ಕನೆಕ್ಷನ್ಗೆ ಏನಂತ ಕರಿತೀವಿ? ಲಿಂಕೇಜ್ ಅಂತ ಕರಿತೇವೆ. ಸೊ ದೇ ಆಸ್ಕ್ಡ್ ದ ಡಿಫರೆನ್ಸಸ್ ಬಿಟ್ವೀನ್ linkage and recombination. Can anyone tell me what do you mean by recombination? Try to tell you in your own words. I'm not forcing you to tell in a more scientific way. I will just rectify your answer if you are wrong. Right? So just try it, guys. What do you mean by recombination? What do you mean by recombination? Anyone? You can switch on your mic and you can tell.
The number of progenies formed during gametogenesis.
The number of progenies formed during what? Gametogenesis.
Yes, ma'am.
Okay. Fine. Okay. Well tried by Deepak. Swati also suggested some answer like non-parental gene anta. So recombination means what? Combination andre the combination of father and mother's genes is called as recombination. I will let you know the exact definition for it. Here it is. Guys, whenever the question comes from differentiate between, please don't forget to put the column, right? So the linkage, here the genes are located in the same chromosome and inherited to the next generation. Here, recombination, here offsprings are formed during separation of genetic material during crossing over. Crossing over anta ಒಂದು ಕಾನ್ಸೆಪ್ಟ್ ಬರುತ್ತೆ ಯಾವಾಗ? ಸೆಲ್ ಡಿವಿಷನ್ ಆಗುವಾಗ, right? When a part of a chromosome, that is a mother's chromosome, and a part of a father's chromosome will cross over and there will be a exchange of genetic material. So ಅದಕ್ಕೆ ನಾವೇನಂತ ಕರಿತೇವೆ? Recombination anta kariteve. That is not during gametogenesis. We have to specify the word that is crossing over anta. Okay. Next, the linked gene is located near the same chromosome. Genes kalu enu linked agirtave, they all will be we can find them in the same chromosome anta. Next, the recombination takes place in the different chromosome. Recombination alli, as I said you, there will be exchange of material, the genetic material between father's and mother's chromosome. So here we can say the recombination occurs between different chromosome located in any portion. Yaude region allu irಬಹುದು. It might be at the end, it might be in the middle, it might be at the tip. So at any position, the crossing over happens, which brings about recombination. As it is given for 2 marks, it is enough if you write 2 points. Here 2 points, here 2 points. So half marks for each difference. I hope you guys got it. Yes, guys? And please don't forget to write the questions while discussing. Okay, na? Fine. Answers you will get, but don't forget to write the questions. Let us proceed to the next question. Differentiate between homozygous and heterozygous condition. Yes, what do you mean by homozygous and heterozygous? It is very easy to say that, but how will you write it? Yes, anyone can try this. Yes, my dear YouTube students, you can answer through your comment box. Differentiate between homozygous and heterozygous. Let me ask you something. So I have written certain genotypes here. So I have written T and T, whether this is homozygous or heterozygous. Okay, good. Deepak answered as homo andre same alleles galirtave. Hetero means a different alleles. Very good. Yes. Identical alleles anta. So identical alleles idre yav ಏನಂತ ಕರಿತೇವೆ? Which condition we will call it as? If both the alleles are identical, ಎರಡು ಅಲೀಲ್ಗಳು ಸೇಮ್ ಇದ್ದಾಗ what we will call that condition as? Be fast guys. Homozygous. Very good. Swati and Deepak are active today. Good. So homozygous condition alva. Next. So here we have T and small letter t. What this condition is referred as? Be fast. Prithvi is here. Vinushri, Swati, Ayyamma, Sita. Heterozygous condition, right? So when the alleles are non-identical, alleles galu non-identical, same ilde hodre, then it is called as heterozygous condition. So you know the concept. ಇದರಿಂದ ಗೊತ್ತಾಯ್ತು ನಿಮಗೆ ಕಾನ್ಸೆಪ್ಟ್ ಗೊತ್ತಿದೆ ಅಂತ. Then how we have to write this answer ಅಂತ ಬಂದಾಗ, here it is. Sometimes the question might be asked for 3 marks. ಕೇಳಿದ್ರೆ 5 marks ಕೇಳ್ಬಿಟ್ರೆ. So that is why this table is given. So yeah, Prithvi, can you read the first 3 differences? Prithvi, can you read? Anushri, can you, sorry, Vinushri, can you read? No response. ಏನಂತ ತಿಳ್ಕೋಬೇಕು ನೋ ರೆಸ್ಪಾನ್ಸ್ ಅಂದ್ರೆ? ಸುಮ್ನೆ ವಿಡಿಯೋ ಆನ್ ಮಾಡ್ಬಿಟ್ಟು ಎಲ್ಲೋ ಹೋಗ್ಬಿಟ್ಟಿದ್ದೀರಾ? ಸ್ವಾತಿ, ಕ್ಯಾನ್ ಯು ರೀಡ್? Sita, can you read? Okay, Swati, you can read. Yes, please, Swati, proceed. Switch on your mic and proceed. Sita, can you read?
Yes, ma'am.
Yeah, sure.
Homozygous, a genetic makeup that consists of 2 identical alleles of a gene for a trait in a diploid organisms. Heterozygous, a genetic makeup that consists of 2 different alleles of a gene for a trait in a diploid organisms. Homozygous, genotypes can be written as AA or small letter a. Heterozygous, genotype can be written as capital A, small a. Homozygous produces single type of gametes. Heterozygous produces 2 types of gametes. Homozygous, 2 phenotypes are the expressed. Heterozygous, 1 phenotype is expressed.
Okay.
Homozygous, self-breeding produces the same trait over the generations. Heterozygous, self-breeding produces a combination of different traits. Homozygous do not show extra effects. Heterozygous shows hybrid effects. Homozygous present in true breeds. Heterozygously present in crossbreeds. Homozygous dominant and homozygous recessive are the 2 types. Heterozygous complete dominance, incomplete dominance, or codominance are the types.
Okay, thank you, Sita. Thank you, Sita. May I know your college name?
Seetama College, ma'am, government, Danagere.
Good. Thank you. So
Thank
here,
you.
homozygous and heterozygous condition for our this one question, that is which is given for 2 marks. It is okay if you write till here. Which one? Till this one. Because ನಾನು ಹೇಳ್ದಾಗೆ ನೀವು 2 ಪಾಯಿಂಟ್ಸ್ ಬರೆದ್ರೆ this homozygous and heterozygous is a what we can say simple concept. ಸೊ ಅದನ್ನ 2 ಪಾಯಿಂಟ್ಸ್ ಬರೆದಾಗ that concept will not go in deep. ಅವ್ರಿಗೆ ಗೊತ್ತಾಗುತ್ತೆ ನಿಮ್ಗೆ ಅಷ್ಟು ಓನ್ಲಿ ಡೆಫಿನಿಷನ್ ಗೊತ್ತು ಅಂತ. ಸೊ ದೇ ಹ್ಯಾವ್ ಟು ನೋ, ಯಾರು ಕರೆಕ್ಷನ್ ಮಾಡ್ತಾರೋ ಅವ್ರಿಗೆ ಗೊತ್ತಿರಬೇಕು ನಿಮಗೆ ನಾಲೆಜ್ ಇದೆ ಅದರ ಬಗ್ಗೆ ಅಂತ. ಸೊ ನಿಮ್ಮ ಪೇಪರ್ ಅಲ್ಲಿ ನಿಮ್ಮ ನಾಲೆಜ್ ರಿಫ್ಲೆಕ್ಟ್ ಆಗಬೇಕು. ದಟ್ ಈಸ್ ವೈ ಇಫ್ ಯು ರೈಟ್ ದೀಸ್ 4 points na, it will be enough. So first one, as Deepak and Swati answered at first, the homozygous will be consisting of identical alleles. Heterozygous will be consisting of 2 different alleles. Genotypes ಯಾವ ರೀತಿ ಬರಿತೀವಿ ಅಂತ ಬರೆಯೋದು. Next, produces single type of gametes. ಯಾವ ರೀತಿ? Suppose if you consider the cross between tall and dwarf. So tall plant to tall gametes ಅನ್ನ ಮಾತ್ರ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡುತ್ತೆ. ಡಾರ್ಫ್ ಪ್ಲಾಂಟ್ ಟು ಡಾರ್ಫ್ ಗ್ಯಾಮೆಟ್ಸ್ ಅನ್ನ ಮಾತ್ರ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡುತ್ತೆ. ಸೊ ಸಿಂಗಲ್ ಟೈಪ್ ಆಫ್ ಗ್ಯಾಮೆಟ್ಸ್. ಬಟ್ ಇನ್ ಹೆಟರೋಸೈಗಸ್ ಕಂಡೀಷನ್ it produces 2 types of gametes, right? Next, 2 phenotypes are expressed, 1 phenotype is expressed. So illi 2 phenotypes are expressed anta. Tall andre tall express agatte, dwarf andre dwarf express agatte. But illi when it is a heterozygous condition TT, only tall will be expressed. Yakandre tallness is a dominant one, right? So that you can write. Okay, so like this you have to write the answer. Let us proceed to the next question. I request everyone to be interactive. Interactive ilde hodre, respond ಮಾಡದೆ ಹೋದ್ರೆ ಗೊತ್ತಾಗಲ್ಲ. Like if you can't read, na, you just message like you have a problem, audio issue or something else. ನೀವು ಏನು ರೆಸ್ಪಾಂಡೇ ಮಾಡಲಿಲ್ಲ ಅಂದ್ರೆ ಹೆಸರು ಕರೆದ್ರೂ ರೆಸ್ಪಾಂಡ್ ಮಾಡಲ್ಲ. If I ask the question, I ask to read and respond ಮಾಡಲಿಲ್ಲ ಅಂದ್ರೆ we feel that you are not here, you are not watching the session, you are somewhere, simply switched on the mobile anta. Okay, next question, a very interesting, simple and most asked question. What is sickle cell anemia and why RBCs become sickle shaped in this condition? Anyone? Sickle cell anemia andre ಏನು ಅಂತ. ಮತ್ತೆ RBC become sickle shaped in this condition. Why? Yes. So we know that our RBCs are there, no? Sorry. So our RBCs, they have a shape of, they are biconcave in shape. Okay. Why it should be biconcave in shape? What is the function of RBC, guys? Yes, my dear future doctors and scientists, let me know what do you mean by, what is the function of RBCs? RBC blood cell gottu. What is the function of it? So its function is to carry the oxygen, alva? Because it has a pigment named hemoglobin, right? So if the shape is like this, ಯಾಕೆ biconcave ಇರಬೇಕಂದ್ರೆ, if the shape is like this, the surface area will be more. ಅಥವಾ ನಾವು ವಿಸ್ತೀರ್ಣ ಅಂತ ಏನ್ ಹೇಳ್ತೇವೆ, the surface area will be more, so that it can take many oxygen atoms or molecules along with it, right? That is why RBCs will be biconcave in shape. No doubt hemoglobin, ಒಂದು hemoglobin 4 oxygen ಅನ್ನ ತೆಗೆದುಕೊಂಡು ಹೋಗುತ್ತೆ ಅಲ್ವಾ? ಸೊ ಬೈಕಾಂಕೇವ್ ಶೇಪ್ ಇದ್ದಾಗ ಹಿಮೋಗ್ಲೋಬಿನ್ಗೆ ಜಾಸ್ತಿ ಆಕ್ಸಿಜನ್ ತೆಗೆದುಕೊಳ್ಳೋ ಹಾಗೆ ವಿಸ್ತೀರ್ಣ, ದಟ್ ಸರ್ಫೇಸ್ ಏರಿಯಾ ಸಪೋರ್ಟ್ಸ್ ಇಟ್. ಐ ಆಮ್ ಸಾರಿ. ಬಟ್ ಅ ಕಂಡೀಷನ್ ಕಾಲ್ಡ್ ಸಿಕ್ಕಲ್ ಸೆಲ್ ಅನೇಮಿಯಾ ಅಂತ ಏನು ಹೇಳ್ತೀವಿ, ದ ಜೆನೆಟಿಕ್ ಡಿಸಾರ್ಡರ್, ಇಟ್ ಮೇಕ್ಸ್ ದ RBC ಆಸ್ ಅ ಸಿಕ್ಕಲ್ ಶೇಪ್, ಅಂದ್ರೆ ಸಿ ಶೇಪ್ ಈ ತರ. ಸೊ ವೆನ್ ಯು ಸಿ ದ ಶೇಪ್ ಇಟ್ ಸೆಲ್ಫ್, ಈ ಸರ್ಫೇಸ್ ಏರಿಯಾ ಏನಿದೆ, ದ ಸರ್ಫೇಸ್ ಏರಿಯಾ ಅಥವಾ ಒಳಗಿನ ವಿಸ್ತೀರ್ಣ ಏನಿದೆ ಅದು ಕಡಿಮೆ ಇದೆ. ಸೊ ಆಬ್ವಿಯಸ್ಲಿ ದ ಟ್ರಾನ್ಸ್ಪೋರ್ಟೇಷನ್ ಆಫ್ ಆಕ್ಸಿಜನ್ ವಿಲ್ ಬಿ ವೆರಿ ಲೆಸ್. ಆಕ್ಸಿಜನ್ನೇ ಇಲ್ಲ ಅಂದಮೇಲೆ ಮೇನ್ ಬಾಡಿ ಪಾರ್ಟ್ಸ್ ಲೈಕ್ ಬ್ರೈನ್, ಹಾರ್ಟ್ ಇಸ್ ಲೈಕ್ ತುಂಬಾ ಡಿಫಿಕಲ್ಟ್ ಆಗತ್ತೆ ಅದು ಫಂಕ್ಷನ್ ಮಾಡೋದು ಅಲ್ವಾ? ಸೊ ಲೆಟ್ ಅಸ್ ಸಿ ಹೌ ಟು ರೈಟ್ ದಿಸ್ ಆನ್ಸರ್. ಎಸ್, ಸ್ವಾತಿ, ಕ್ಯಾನ್ ಯು ರೀಡ್ ಇಟ್?
Sickle cell anemia is caused due to the point mutation, because of which is the B globin chain of hemoglobin molecules, the glutamic acid CLUG, replaced by the valine. This results in oxygen stress. Under this condition, the RBC lose their circular shape, polygenerize and become agglutinated and sickle shaped.
Thank you, Swati. May I know your college name?
Government College Tekilkote in district Ballari.
Great, thank you. So sickle cell anemia anta ಹೇಳ್ತಿದಾರಲ್ವಾ? So it is caused due to, it is a genetic disorder caused by the point mutation, because of which B globulin chain of hemoglobin molecule. Hemoglobin ಗೆ 2 ಚೈನ್ ಇರುತ್ತೆ, ಅದರಲ್ಲಿ ಒಂದು ಚೈನ್ ಹೆಸರು B globulin ಅಂತ. ಸೊ ಆ ಮಾಲಿಕ್ಯೂಲ್ chain of hemoglobin molecule, the glutamic acid, ಆ ಮಾಲಿಕ್ಯೂಲ್ ಚೈನ್ ಅಲ್ಲಿ glutamic acid ಅನ್ನೋ ಅಮೈನೋ ಆಸಿಡ್ ಏನಿದೆ, ಇಟ್ ಈಸ್ ರೀಪ್ಲೇಸ್ಡ್ ಬೈ ವ್ಯಾಲಿನ್. ಸೊ ಅದು ವ್ಯಾಲಿನ್ ಇಂದ ರೀಪ್ಲೇಸ್ ಆದಾಗ ಏನಾಗುತ್ತೆ, ದ ಶೇಪ್ ಆಫ್ ದ ಆರ್ ಬಿ ಸಿ changes into the sickle shape, as you can see here, alva. So as you can see in this diagram, right, sickle shape ಆದಾಗ you can see here guys, normal shape ಇದ್ದಾಗ see the blood flow, ಇದು blood vessel, ಇದು see the blood flow, it is easy. when sickle shaped occurs ಅಲ್ವಾ, ಇಲ್ಲಿ ನೋಡಿ sickle shape ಆದಾಗ blood vessels ಅಲ್ಲಿ ಈ ರೀತಿ ಬ್ಲಾಕ್ ಆಗ್ಬಿಡತ್ತೆ. when there is a block, there is no transportation, the circulation becomes a hectic job, right? So that is why sickle shaped anemia ಅನ್ನೋದು, it is a most dangerous and a lethal disease. Like this we have to write the answer. ಏನ್ ಕೊಟ್ಟಿದ್ದಾರೆ, what is sickle cell anemia? It is a genetic disorder caused by point mutation where glutamic acid is replaced by valine ಅಂತ ಬರೋದು. what it causes, ಏನ್ ಮಾಡತ್ತೆ ಅದು, it changes the shape of RBC into a sickle shape ಅಂತ ಬರಿಬಹುದು. yes, why RBC becomes sickle shaped ಅಂದ್ರೆ ಇದರ ಸಲುವಾಗಿ, ಯಾವ್ದ್ರ ಸಲುವಾಗಿ, ಗ್ಲುಟಮಿಕ್ ಆಸಿಡ್ ಇದ್ದಿದ್ದೇನಾಗಿರುತ್ತೆ, ಇಟ್ ಈಸ್ ರೀಪ್ಲೇಸ್ಡ್ ಬೈ ವ್ಯಾಲಿನ್. ಇದು ಇಟ್ ರಿಸಲ್ಟ್ಸ್ ಆಕ್ಸಿಜನ್ ಸ್ಟ್ರೆಸ್ ಅನ್ನ ತರತ್ತೆ, ಅಂಡರ್ ದಿಸ್ ಕಂಡೀಷನ್ ದ RBC become sickle shaped. like this we have to write. 2 marks ಗೆ ಕೊಟ್ರೆ ಇದನ್ನಷ್ಟೇ ಬರೆಯೋದು ಅಡ್ಡಿ ಇಲ್ಲ, in the words. just a minute. okay, 2 marks ಗೆ ಕೊಟ್ರೆ it is enough if you write this, but if they asked same for a 3 marks, it is good if you write the diagram ಇಂದ ನೀವು ಅದನ್ನ ರಿಪ್ರೆಸೆಂಟ್ ಮಾಡಿದ್ರೆ. okay, fine. 3 marks ಕೊಟ್ಟಾಗ if you represent this diagram, then it will be helpful for you. so here hemoglobin ಅಂತ ಇದೆ ಅಲ್ವಾ, hemoglobin, heme is what, a iron group. ದೀಪಕ್, heme is a iron group. globin ಅನ್ನೋದು ಒಂದು ಪ್ರೋಟೀನ್. so globin protein will be having the chains. okay, globin protein will be having the chain beta globulin, ಅದು ಬಿ ಅಲ್ಲ, beta globulin chain. so ಈ beta globulin chain, it is made up of, we know that proteins are made up of amino acids ಅಲ್ವಾ, proteins are what, amino acid chain proteins ಗಳಾಗ್ತವೆ. so ಇಲ್ಲಿ proteins or amino acid, what is there, one of the amino acid named glutamic acid. ನಾರ್ಮಲ್ ಆಗಿ ಗ್ಲುಟಮಿಕ್ ಆಸಿಡ್ ಇರಬೇಕು ಯಾವುದರಲ್ಲಿ, ಬೀಟಾ ಗ್ಲೋಬುಲಿನ್ ಚೈನ್ ಅಲ್ಲಿ. that glutamic acid is replaced by a amino acid called valine. ಗ್ಲುಟಮಿಕ್ ಆಸಿಡ್ ಬದಲು ಅಲ್ಲಿ ವ್ಯಾಲಿನ್ ಬಂದಿರುತ್ತೆ, ಯಾಕೆಂದ್ರೆ ಡ್ಯೂ ಟು ದ ಪಾಯಿಂಟ್ ಮ್ಯೂಟೇಷನ್, ಡ್ಯೂ ಟು ಚೇಂಜ್ ಇನ್ ಅ ಸಿಂಗಲ್ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಇಂದ ಅಲ್ಲಿ ವ್ಯಾಲಿನ್ ಬರುತ್ತೆ. ಸೊ ವ್ಯಾಲಿನ್ ಬಂದ್ ಏನ್ ಮಾಡತ್ತೆ, ಇಟ್ ರಿಸಲ್ಟ್ಸ್ ಇನ್ ಆಕ್ಸಿಜನ್ ಸ್ಟ್ರೆಸ್, ಆಕ್ಸಿಜನ್ ನ ಕಡಿಮೆ ಮಾಡತ್ತೆ, ಅಂದ್ರೆ ಆಕ್ಸಿಜನ್ ಕೆಪಾಸಿಟಿ ಅಥವಾ ಟ್ರಾನ್ಸ್ಪೋರ್ಟೇಷನ್ ಅನ್ನ ಕಡಿಮೆ ಮಾಡತ್ತೆ, ವಿಚ್ ಮೇಕ್ಸ್ or which converts the shape of the RBC into sickle shape. I hope it is clear now, Deepak. Is it clear, Deepak? Okay, may I know your college name? Okay, the next question goes like this. Okay, good. Thank you. The next question goes like this. Give the phenotypes of the parental Drosophila. T.H. Morgan demonstrated the linkage experiments, right? So, ಅವರಲ್ಲಿ give the phenotypes of parental Drosophila that produced 1.3% and 37.2% recombinants respectively in the T.H. Morgan dihybrid cross experiment. Yes, my dear YouTube students. ಏನ್ ಕೇಳಿದ್ದಾರೆ? ಈ ಒಂದು ಪರ್ಸೆಂಟೇಜ್ ನೋಡಿ ನಾವು ಭಯ ಬಿದ್ಬಿಡ್ತೇವೆ, ಅಯ್ಯೋ ಇದು ಬೇರೆ ಏನೋ ಕೊಟ್ಟಿದ್ದಾರೆ ಅಂತ. ವಿ ಹ್ಯಾವ್ ಟು ಕ್ಯಾಲ್ಕುಲೇಟ್ ಸಮಥಿಂಗ್ ವಿ ವಿಲ್ ಥಿಂಕ್. ಅದೇ ಈ ರೀತಿ ಎಕ್ಸಾಮಿನೇಷನ್ ಹಾಲ್ ಅಲ್ಲಿ ಕುತ್ಕೊಂಡಾಗ ವಿ ವಿಲ್ ಬಿಕಮ್ ಅ ಸೈಂಟಿಸ್ಟ್ ಆರ್ ಅ ರಿಸರ್ಚ್ ಪೀಪಲ್. ಲೈಕ್ ವಿ ವಿಲ್ ರೈಟ್ ಅವರ್ ಓನ್ ರಿಸರ್ಚ್. ಇಟ್ ಈಸ್ ಗುಡ್, ಬಟ್ ಇಟ್ ಈಸ್ ನಾಟ್ ಅ ಆಕ್ಚುಯಲ್ ಎಕ್ಸಾಕ್ಟ್ ಟೈಮ್ ವೇರ್ ಯು ಕ್ಯಾನ್ ರೈಟ್ ಯುವರ್ ರಿಸರ್ಚ್ ವರ್ಕ್. ಸೈಂಟಿಸ್ಟ್ ಆದ್ಮೇಲೆ ಬರೀರಿ ನೀವು. ಬಟ್ ಇಲ್ಲಿ 1.3% ಇದ್ರಲ್ಲಿ ಏನಿಲ್ಲ, ಸಿಂಪಲ್, ವೆರಿ ಸಿಂಪಲ್ ಕ್ವಶನ್ ಕೇಳಿದ್ದಾರೆ. ಏನ್ ಕೇಳಿದ್ದಾರೆ? ಈಗ T.H. ಮಾರ್ಗನ್ ಅವರು ಡೈಹೈಬ್ರಿಡ್ ಕ್ರಾಸ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಅನ್ನ ಮಾಡ್ತಾರೆ. ಸೊ ಅವರಿಗೆ ಸಿಕ್ಕಂತಹ ರಿಸಲ್ಟ್ ಏನು? 1.3% ಅವರಿಗೆ ಒಂದಿಷ್ಟು ರೆಡ್ ಐಡ್ ಫ್ಲೈ ಸಿಗ್ತವೆ. 37.2% ವಿಲ್ ಬಿ ಆಫ್ ಪೇರೆಂಟಲ್. ಸೊ ಇದರಲ್ಲಿ ಈ ರೀತಿ ಸಿಕ್ಕಂತಹ ಪ್ರೋಜೆನೀಸ್ ನ ಹಿಂದಿನ ಜನರೇಷನ್ ಅಂದ್ರೆ ಪೇರೆಂಟ್ಸ್ ಯಾವ ರೀತಿ ಇದ್ರು, ಅಂಡ್ ಇವನ್ ದೇ ಆಸ್ಕ್ಡ್ ಫಿನೋಟೈಪ್ಸ್. ವೆರಿ ಈಸಿ ಕ್ವಶನ್. ಕ್ಯಾನ್ ಎನಿವನ್ ಟ್ರೈ ದಿಸ್? ಯಾರಾದ್ರೂ ಟ್ರೈ ಮಾಡ್ಬೋದಾ? ಎಸ್ ಎಸ್ YouTube students. 1:2:1. They did not ask phenotypic ratio. They asked phenotypes. ಯಾವ ರೀತಿ ಇತ್ತು? What about their mother? What about their father? ಆ ರೀತಿ ಅಂತ. So here we have to answer like this. In T.H. Morgan's Drosophila dihybrid cross experiment, the parental Drosophila that produced 1.3% and 37.2% recombinants are heterozygous for both traits being studies. ಅವರು ಎರಡು ಟ್ರೇಟ್ಸ್ ಅಥವಾ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಅನ್ನ ಡಿಸ್ಕಸ್ ಮಾಡ್ತಾರೆ. ಯಾವನ್ನ? ಒಂದು ರೆಡ್ ಐಡ್ ಅಂಡ್ ಇನ್ನೊಂದು ರಿಗಾರ್ಡಿಂಗ್ ದ ವಿಂಗ್ಸ್. ಓಕೆ, ರಿಗಾರ್ಡಿಂಗ್ ದ ವಿಂಗ್ಸ್. ಸೊ ಎರಡರಲ್ಲೂ ಇರುವ ಎರಡು ಕ್ಯಾರೆಕ್ಟರ್ನ ಹೆಟರೋಸೈಗಸ್ ಕಂಡೀಷನ್ when he crossed both the parents, both the parents were heterozygous in both the characters, that is in the wing character as well as in the eye character. So ಆ ರೀತಿ ಇದ್ದಾಗ we got 1.3 and 37.2% recombinants ಅಂತ ಹೇಳ್ತಾರೆ. Fine, let us proceed to the next question then. Okay, very easy question. List and guys, ಈ ಒಂದು ಕ್ವಶನ್ ಏನಿದೆ ಅಲ್ವಾ, this one, it has been asked for in MQP this year only, 2023-24 model question paper ಅಲ್ಲಿ ಈ ವರ್ಷದ model question paper which is released by the board. In that model question paper, this question has been asked and the answer is very simple. As I said you, in both the character, the parents will be heterozygous in condition. Okay, let us proceed to the next question. List the features of Drosophila melanogaster which makes it suitable for genetic experiments. Yes, why they select Drosophila melanogaster to do genetic experiments? Yes guys, ತುಂಬಾ ಈಸಿ ಕ್ವಶನ್ ಅಲ್ವಾ? You can try this. List the features of Drosophila melanogaster, which makes it suitable for genetic experiments. Very good. Deepak HS answered as they grow on synthetic medium. Fine. Number of progenies will be more accurate. Very good. Yes. What about others? Shall I ask individually? Be fast. Use your chat box. Lifetime is short. That is about 16 days. Okay. Guys, if you can't interact with the mic, ನನ್ನ ಲ್ಯಾಂಗ್ವೇಜ್ ಸರಿ ಇಲ್ಲ, ನನಗೆ ಮಾತಾಡಕ್ ಬರಲ್ಲ, ನನಗೆ ಭಯ ಆಗತ್ತೆ, ನಾನು ಹೇಳಿದ ಮೇಡಮಿಗೆ ಅರ್ಥ ಆಗಲ್ಲ, ನಾನು ಯಾವಾಗ್ಲೂ ತಪ್ಪೇ ಹೇಳ್ತೀನಿ, ಆ ರೀತಿ ಏನಾದ್ರೂ ನಿಮಗೆ ನೆಗೆಟಿವ್ ಥಾಟ್ಸ್ ಇದ್ರೆ ಯು ಕ್ಯಾನ್ ಯೂಸ್ ದ ಚಾಟ್ ಬಾಕ್ಸ್ ಟು ಆನ್ಸರ್. ಎಸ್, ಬಿ ಫಾಸ್ಟ್. Chromosome number is 2n is equal to 8. Okay, Swati said it. Good. Deepak answered well. Swati also tried. Good. Yes. What about others? Sita, Prithvi, Ayyamma, Vinushri. One more student is there. I don't know the exact name. Kannadiga anta ide. A single mating can produce large number of progenies. Answered by Sita. Good. Yes. What about others? Yes, Ayyamma, you want to tell something?
They completed their life cycle with 15 days.
Okay, they complete their life cycle within 15 days. Good. Okay, let us proceed to the next answer. See, following are the features. And one more thing, guys. ನೀವು ಈಗ ಯೂಶ್ವಲಿ ಬಯಾಲಜಿ, ಫಿಸಿಕ್ಸ್, ನಮ್ಮ ಸೈನ್ಸ್ ಸ್ಟ್ರೀಮ್ನವ್ರಿಗೆ ಏನು ಗೊತ್ತಾ, ನಾವ್ ಯಾಕೆ ಸುಮ್ಮನೆ ಲ್ಯಾಂಗ್ವೇಜಿಗೆ ಫೋಕಸ್ ಮಾಡಬೇಕು ಅಂತ. ದಟ್ ಓವರ್ ಕಾನ್ಫಿಡೆನ್ಸ್ ವಿಲ್ ಬಿ ದೇರ್. Okay, we can see it in a microscope. Okay, good, Sita. That overconfidence will be there. But answers ಬರೆಯುವಾಗ ಒಂದು ಮೆಥಡ್ ಅನ್ನ ನಾವು ಫಾಲೋ ಮಾಡಲೇಬೇಕು, ರೈಟ್? ಸೊ ಹಿಯರ್ ಡೈರೆಕ್ಟ್ಲಿ ಡೋಂಟ್ ಗೋ ಟು ದ ಪಾಯಿಂಟ್ಸ್. ಇಲ್ಲಿ ನೋಡಿ ಎಷ್ಟು ಚೆನ್ನಾಗಿ ಬರೆದಿದ್ದಾರೆ. ದ ಆನ್ಸರ್ ಹ್ಯಾಸ್ ಬೀನ್ ಡೌನ್ಲೋಡೆಡ್ ಫ್ರಮ್ ದ ಆನ್ಸರ್ ಕಾಪಿ ಆಫ್ ದ ಟಾಪರ್ ಸ್ಟೂಡೆಂಟ್. ಓಕೆ, ಸೊ ವಿಚ್ ಐ ಗಾಟ್ ಇನ್ ದ ವೆಬ್. ಫೈನ್. ಸೊ ಇಲ್ಲಿ following features of Drosophila make it ideal for genetical studies. See the wordings. ನಮಗೆ ಇಂಗ್ಲಿಷ್ ಗೊತ್ತಿಲ್ಲ ಮೇಡಮ್, ನಾವ್ ಹೇಗ್ ಬರಿಬೇಕು ಅಂತ ಅಂದ್ರೆ ಗೊತ್ತಿರೋದೇ ಬೇಡ, ಅಲ್ಲೇ ಕೊಟ್ಟಿದ್ದಾರೆ. ಲಿಸ್ಟ್ ದ ಫೀಚರ್ಸ್. ಫಾಲೋಯಿಂಗ್ ಆರ್ ದ ಫೀಚರ್ಸ್ ಆಫ್ ಡ್ರೆಸೋಫಿಲಾ. ಫಾಲೋಯಿಂಗ್ ಅಂತ ಒಂದು ವರ್ಡ್ ನಿಮಗೆ ಗೊತ್ತಿದ್ರೆ ಸಾಕು. ಫಾಲೋಯಿಂಗ್ ಆರ್ ದ ಫೀಚರ್ಸ್ ಆಫ್ ಡ್ರೋಸೋಫಿಲಾ ಮೆಲಾನೋಗಾಸ್ಟರ್ ವಿಚ್ ಮೇಕ್ಸ್ ಇಟ್ ಸೂಟೇಬಲ್ ಫಾರ್ ಜೆನೆಟಿಕ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ಸ್ ಅಂತ ಬರೋದು. When you write these points, alva, then it will be nice, right? It will be nice. Okay, representation chanag ansutte. So the first here, it can be easily grown on simple synthetic medium. As Deepak said it, it is a right. It completes its life cycle within 2 weeks. That is 15 days anta Sita answered it, Ayyamma answered it, and even Deepak. A single mating produces large number of offsprings. There is clear-cut sexual dimorphism. Very important point. ಈ ಎಲ್ಲದರಲ್ಲಿ ಇಂಪಾರ್ಟೆಂಟ್ ಪಾಯಿಂಟ್ ಇದು. Sexual dimorphism ಅಂದ್ರೆ we can easily identify which is a male Drosophila melanogaster, which is a female Drosophila melanogaster. ಈಗ ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಹೌಸ್ ಫ್ಲೈ ಅಂತ ತಿಳ್ಕೊಳ್ಳಿ. ನೊಣ ನಿಮ್ ಮನೇಲಿ ಬರುತ್ತೆ ಅಥವಾ ನಿಮ್ ಕೈ ಮೇಲೆ ಕೂತ್ಕೊಂಡಿದೆ. ಕ್ಯಾನ್ ಯು ಜಸ್ಟ್ ರೆಕಗ್ನೈಸ್ ವೆದರ್ ಇಟ್ ಇಸ್ ಅ ಮೇಲ್ ಆರ್ ಅ ಫೀಮೇಲ್ ಈಸಿಲಿ? No, but we can exactly what we can say identify which is a male one and which is a female one. It has many heredity variation that can be easily observed with low power microscope. ನಮಗೆ ಇದಕ್ಕೆ ಎಲೆಕ್ಟ್ರಾನ್ ಮೈಕ್ರೋಸ್ಕೋಪೇ ಬೇಕಂತ ಏನಿಲ್ಲ. ಲೋ ಪವರ್ ಮೈಕ್ರೋಸ್ಕೋಪ್ ಇಂದನೂ ಕೂಡ we can see or we can study the structure of it, right? So these are the points. Well tried, my dear students. Let us proceed to the next question. Yeah. What is test cross? Write its significance. Anyone, be fast. What is test cross? Test cross andre enu? Adra mahatva enu anta? Yes, my dear YouTube students. What is test cross? Write its significance. Try it. Be fast. ತುಂಬಾ ಇಂಟ್ರೆಸ್ಟಿಂಗ್ ಚಾಪ್ಟರ್ ಇದು. ಯಾವ ರೀತಿ ಕ್ವಶನ್ಸ್ ಕೇಳಬೇಕು, ಕ್ವಶನ್ಸ್ ಕೇಳೋದು ಮುಗಿತೀನಿ, ಎಕ್ಸಾಮ್ ಹತ್ರ ಬಂತು. You have to answer, right? So Deepak answered as kind of back cross between recessive parents. Partially correct. Well tried. Well done. Kind of back cross between recessive parents. ಒಂದು recessive parent ಇರತ್ತೆ, ಇನ್ನೊಂದು in the cross one will be recessive parent, the other one will be what? Yes, anyone? Again, dominant. Okay, fine. So here what happens, guys? The crossing of F1 progeny, first generation ಏನಿರತ್ತಲ್ವಾ, so the crossing of first generation, that is with a double homozygous recessive parental progeny. For example here, yes Sita, I will come to your point. ಈಗ F1 progeny ಮೆಂಡಲ್ monohybrid cross ಅಲ್ಲಿ ಏನ್ ಬಂತು, heterozygous tall ಬಂತು. Crossing this F1 generation with the homozygous recessive parent, okay, this is called as test cross. Okay, see, progeny is done in order to determine, ಯಾಕೆ ಮಾಡ್ತೇವೆ, whether the progeny is homozygous or heterozygous. F1 ನಲ್ಲಿ ಬಂದಿರುವ progeny homozygous ಇದೆನಾ ಅಥವಾ heterozygous ಇದೆನಾ ಅದನ್ನ ಐಡೆಂಟಿಫೈ ಮಾಡಕ್ಕೆ we will do this, what, test cross ಅನ್ನ ಮಾಡ್ತೇವೆ. Very good. Swati and Sita said this. Fine. Significance ಗೆ ಬಂದಾಗ first one is determine the genotype of the F1 progeny. On the basis of the results obtained in the ratio of the offspring, it can be predicted that parent is having which genotype. So ಈಗ ನಮಗೆ F1 generation ಅಲ್ಲಿ TT ಅಂತ ಬಂದಾಗ ನಾವು ಪ್ರೆಡಿಕ್ಟ್ ಮಾಡಬಹುದು, ಊಹಿಸಬಹುದು, ಏನಂತ, ಪೇರೆಂಟ್ಸ್ ಅಲ್ಲಿ ಟಾಲ್ನೆಸ್ ಇದೆ, ಪೇರೆಂಟ್ ಅಲ್ಲಿ ಡಾರ್ಫ್ನೆಸ್ ಇದೆ ಅಂತ ಊಹಿಸಬಹುದು. Then if all the offsprings are of same phenotype, then the parent is homozygous dominant. So ಎಲ್ಲ ಒಂದು ಆಫ್ಸ್ಪ್ರಿಂಗ್ಸ್ ಗಳು ಫಿನೋಟೈಪ್ ಟಾಲೇ ಇತ್ತಂದ್ರೆ ದೆನ್ ನಾವೇನಂತ ಅನ್ಕೋಬೇಕು, ಪೇರೆಂಟ್ ಹೋಮೋಸೈಗಸ್ ಕಂಡೀಷನ್ ಅಲ್ಲಿ ಇದ್ದಾರೆ. If there is 1:1 in the phenotype, ಒಂದು ಟಾಲ್ ಇತ್ತು ಒಂದು ಡಾರ್ಫ್ ಇತ್ತು ಅಂತ ಅಂದ್ರೆ we should understand that it is in heterozygous condition, right? So what is the question? What is test cross? Write its significance anta bandaga like this we have to write. Understood everyone? Guys, in the revision class actually my part should be very less, your part should be more, but now it is here, it is becoming ulta. My part is getting more and your participation is getting less. Okay, let us proceed to the next question. What is meant by Mendel's law of independent assortment? Very easy. You have to just explain, describe the law. Law ide alva, adanna describe ಮಾಡಬೇಕು. What is meant by Mendel's law of independent assortment? Anyone who is going to answer this first? Yes, fast. Use your chat box, use your mic. Yes, my dear YouTube students. Yes, guys, very easy question, direct question alva. Mendel's law of independent assortment andre ಏನು? You have to state it. See here. Mendel's law of independent assortment states that in ಹೇಳುತ್ತೆ ಅದು the alleles of two different genes get sorted into gametes independently. Yes, Deepak, I will come to your answer. Of one another. In other words, the allele of a gamete received for one gene does not influence the allele received for another gene. So Deepak answered it. When two pairs of traits are combined in a hybrid, segregation of one pair of character is independent of the other pair. Okay, this is also correct. See here, according to Mendel's law of, okay, Sita also answered it. So ಇಲ್ಲಿ ನೋಡಿ, yellow and round seeds are taken, green and wrinkled seeds are taken. And in the progeny, that is in a F1 generation, we got yellow and round seeds. Next, in the F2 generation, we got yellow and round seeds, round and green seeds, green and yellow and wrinkled seeds. So new characters bantu, old characters bantu. ಇದರಿಂದ ನಮಗೇನು ಗೊತ್ತಾಯ್ತು? Yellow color of a seed is not dependent on the shape, that is round shape of the seed. Green color of the seed is not dependent on the wrinkled shape of a seed. So both the characters will express independently without any mixing up, without depending on each other. So this is called as Mendel's law of independent assortment. Rohit Raju also answered it. The alleles of two or more different genes are assorted. Good. So here, if it is asked for 2 marks, 2 ಅಂಕಗಳಿಗೆ ಅಥವಾ ಎರಡೇ ಮಾರ್ಕ್ಸ್ಗೆ ಕೇಳಿದ್ರೆ, it is good if you write this. ಇಲ್ಲಿವರೆಗೂ ಬರೆದ್ರೆ ಸಾಕು. The same thing if it is asked for 5 marks. 5 marksಗೆ ಬಂದಾಗ ಏನದು, what is Mendel's law of independent assortment? Explain, elaborate ಅಂತ ಏನಾದ್ರೂ ಹೀಗೆ ಕೇಳ್ತಾರೆ. ಅವಾಗ ಏನ್ ಮಾಡ್ಬೇಕು? You have to solve the dihybrid cross ಇನ ಎಕ್ಸಾಂಪಲ್ ಅನ್ನ ಸಾಲ್ವ್ ಮಾಡಿ you have to state the law of independent assortment. I hope it is clear now. Yes, guys. Yes, my dear YouTube students. Okay, good. Fine, Kirtana. Let us proceed to the next question. Okay, Rohit. Then the next question goes like this. Why a cut results in non-stop bleeding in hemophilia? Hemophilia ಅಂತ ಒಂದು ಕಂಡೀಷನ್ ಬರುತ್ತೆ. ಆ ಒಂದು ಹೀಮೋಫೀಲಿಯಾ ಇದ್ದ ವ್ಯಕ್ತಿಗಳಿಗೆ the bleeding will not stop at all. Why? Next question. The possibility of a female becoming hemophilic is extremely rare. Female ಅಲ್ಲಿ hemophilia ಬರೋದು ತುಂಬಾ ರೇರ್ ಅಂತೆ. ಯಾಕೆ? These 2 question has been asked for 2 marks. ಅಂದ್ರೆ 1 ಮಾರ್ಕಿಗೆ 1 question ತರ. Yes, who want to answer this? Why a cut results in non-stop bleeding in hemophilia? Yes, guys. Yes, YouTube students, good. Be interactive. Please answer in the comment box. Okay, Deepak answered it. A single protein that is a part of a cascade of proteins involved in clotting of a blood is affected. Okay, fine. What about others? Yeah, be fast. Why cut results in non-stop bleeding? Hemophilia ಇರುವ ವ್ಯಕ್ತಿಗೆ non-stop bleeding ಯಾಕಾಗತ್ತೆ? Here it is. So, hemophilia is usually an inherited bleeding disorder. First, we have to write the introduction. ಏನದು? Bleeding disorder, blood related disorder ಅದು, which is inherited. ಅಂದ್ರೆ ವಂಶದಿಂದ ವಂಶಕ್ಕೆ, ಪೀಳಿಗೆಯಿಂದ ಪೀಳಿಗೆಗೆ ಬರುವಂತಹ ಒಂದು disorder ಅದು. Right? Fine. This can lead to spontaneous bleeding. ಅವಾಗ ಏನಾಗತ್ತೆ? In which the blood does not clot. ಅಂದ್ರೆ ರಕ್ತ ಹೆಪ್ಪುಗಟ್ಟೋದಿಲ್ಲ ಅಲ್ಲಿ. ಯಾಕೆಂದ್ರೆ our blood will be containing many proteins. ಆ ಪ್ರೋಟೀನ್ಸ್ಗೆ we will call it as clotting factor. ಆ ಪ್ರೋಟೀನ್ಸ್ ಕೆಲಸ ಏನು ಅಂತಂದ್ರೆ to clot the blood. ರಕ್ತವನ್ನ ಹೆಪ್ಪುಗಟ್ಟಿಸೋದು. ಸೊ ಆ ಕ್ಲಾಟಿಂಗ್ ಫ್ಯಾಕ್ಟರ್ಸ್ ಏನ್ ಮಾಡತ್ತೆ? It will help to stop the blood. ಬಟ್ ಯಾವ ಮನುಷ್ಯರಿಗೆ ಅಥವಾ ಯಾವ ವ್ಯಕ್ತಿಗೆ ಹೀಮೋಫೀಲಿಯಾ ಇರತ್ತೆ, ದೇ ವಿಲ್ ಬಿ ಹ್ಯಾವಿಂಗ್ ಲೋ ಲೆವೆಲ್ಸ್ ಆಫ್ ಕ್ಲಾಟಿಂಗ್ ಫ್ಯಾಕ್ಟರ್. ಅವರಲ್ಲಿ ಈ ಪ್ರೋಟೀನ್ಸ್ ಗಳು ಅಥವಾ ಕ್ಲಾಟಿಂಗ್ ಫ್ಯಾಕ್ಟರ್ಸ್ ಗಳೇನಿರತ್ತೆ ಕಡಿಮೆ ಇರತ್ತೆ. ಸೊ ದೇ ವಿಲ್ ಬಿ ಹ್ಯಾವಿಂಗ್ ಸ್ಪಾಂಟೇನಿಯಸ್ ಬ್ಲೀಡಿಂಗ್. ಅವ್ರಿಗೆ ಏನಾದ್ರೂ ಕಟ್ಟಾದ್ರೆ ಕಂಟಿನ್ಯೂಸ್ ಆಗಿ ಬ್ಲೀಡಿಂಗ್ ಆಗತ್ತೆ. ನೆಕ್ಸ್ಟ್, ದೀಸ್ ಡಿಸಾರ್ಡರ್ಸ್ ಎಫೆಕ್ಟ್ ಮೇಲ್ಸ್. ಇದು ಮೇಲ್ ಅಥವಾ ಗಂಡು ಮಕ್ಕಳಿಗೆ ಜಾಸ್ತಿ ಎಫೆಕ್ಟ್ ಮಾಡತ್ತೆ. ಬಿಕಾಸ್ ಯಾಕೆ ಅಂತ ಅಂದ್ರೆ some of you are just copying and you are pasting the answers here. ಹೌದಾ? So you write your own answers. Fine, we will get to know. Chat box ಅಲ್ಲಿ ಆ ಲಿಂಕ್ ತೋರಿಸ್ಬಿಡತ್ತೆ ನೀವು ಎಲ್ಲಿಂದ ಡೌನ್ಲೋಡ್ ಮಾಡಿರ್ತೀರಾ ಅಂತ. We will get to know. ನಾವು we will be having that this na SS na. So fine. So these disorders affect males. ಇದು males ಗೆ ಜಾಸ್ತಿ ಎಫೆಕ್ಟ್ ಮಾಡುತ್ತೆ more often than females because we females will be having 2 X chromosome. ನಮಗೆ 2 X chromosome ಗಳಿರ್ತವೆ. So the gene which is affecting this clotting factor will be present on X chromosome, right? But ನಮಗೇನಾಗತ್ತೆ, ಒಂದು X chromosome ಮೇಲೆ ಈ ಒಂದು ಜೀನ್ ಇದ್ರೆ ಇನ್ನೊಂದು X chromosome ಮೇಲೆ ಇರೋದಿಲ್ಲ. So that is why we will not exhibit or we will not get hemophilia. We will be the carriers. ನಾವು carriers ಆಗಿ ಪರಿಗಣಿಸ್ತಾರೆ ನಮ್ಮನ್ನ, ಫೀಮೇಲ್ಸ್ ಅನ್ನ. And we will not get hemophilia. But male ಅಲ್ಲಿ ಏನಾಗತ್ತೆ, male will be having only 1 X chromosome. ಅಲ್ವಾ? ಸೊ ಆ ಜೀನ್ ಏನಾಗತ್ತೆ, ಇದೇ ಒಂದು X chromosome ಮೇಲೆ ಆ ಜೀನ್ ಏನಾದ್ರೂ ಮ್ಯೂಟೇಟ್ ಆದ್ರೆ, if that gene is affected, see here, any mutation in the factor 8 or 9th gene ಅಲ್ಲಿ ಏನಾದ್ರೂ ಮ್ಯೂಟೇಷನ್ ಆದ್ರೆ which results in hemophilia in the male. ಯಾಕೆಂದ್ರೆ ಮೇಲ್ ಅಲ್ಲಿ ಇರೋದು ಒಂದೇ ಎಕ್ಸ್ ಕ್ರೋಮೋಸೋಮ್, ನಮ್ಮಲ್ಲಿ 2 ಇರೋದ್ರಿಂದ ಒಂದಕ್ಕೆ ಎಫೆಕ್ಟ್ ಆದ್ರೂ ಇನ್ನೊಂದು ಮ್ಯಾನೇಜ್ ಮಾಡತ್ತೆ. ಓಕೆ, ಲೈಕ್ ದಿಸ್ ವಿ ಹ್ಯಾವ್ ಟು ರೈಟ್. ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್ ಗೋಸ್ ಲೈಕ್ ದಿಸ್. ಅ ಮ್ಯಾನ್ ಆಫ್ ಬ್ಲಡ್ ಗ್ರೂಪ್ ಎ, ಮ್ಯಾನ್ ಇದ್ದಾನೆ, ಬ್ಲಡ್ ಗ್ರೂಪ್ ಎ, marries a woman with blood group B. Okay, woman blood group B ಇದೆ, man in a blood group A ಇದೆ. The first child of a couple has O blood group. ಫಸ್ಟ್ ಅಂದ್ರೆ ಒಂದನೇ ಮಗು ಏನಿದೆ, it has a O blood group. What other blood groups are expected in future children? ಮುಂಬರುವ ಮಕ್ಕಳಲ್ಲಿ ಯಾವ ಯಾವ ಬ್ಲಡ್ ಗ್ರೂಪ್ ಗಳ ಮಕ್ಕಳು ಹುಟ್ಟಬಹುದು ಅಂತ ಕೇಳಿದ್ದಾರೆ. Yes, who is going to answer this? Very easy question ಅಲ್ವಾ, ಸಾಲ್ವ್ ಮಾಡಿ ಆನ್ಸರ್ ಮಾಡಬಹುದು. ಟ್ರೈ ಮಾಡಿ, ಪ್ಲೀಸ್ ಟ್ರೈ. ವೆರಿ ಗುಡ್. ದೀಪಕ್ ಆನ್ಸರ್ಡ್ ಆಸ್ A, B, A and B and O. O ಬರ್ದಿಲ್ಲ ಅವರು. ಸ್ವಾತಿ ಆನ್ಸರ್ಡ್ ಆಸ್ ಓನ್ಲಿ A. ಓಕೆ, ವೆಲ್ ಟ್ರೈಡ್, ಗುಡ್. ಸೊ ಹಿಯರ್ ಈ ರೀತಿ ಕ್ವಶನ್ಸ್ ಗಳು ಬಂದಾಗ ನಾವು ಯಾವ ರೀತಿ ಅದನ್ನ ರಿಪ್ರೆಸೆಂಟ್ ಮಾಡಬೇಕು? Okay, Sita answered well. Okay, first what we have to write is the blood group of the man is A, which means the possible genotypes. A blood group idre genotype ಏನಿರುತ್ತೆ? ಒಂದು homozygous condition, ಒಂದು heterozygous condition. Right? Next, likewise, the blood group of women is B anta andaga, which means possible genotypes. One is homozygous, one is heterozygous. Then A, B, AB and O are the possible blood groups of the offspring. So idanna yav reeti baribeku? First we have to put one table like this. Fine. After that, ee reeti bardu. So here we will write parents anta bariyona. Here we will write condition, yava condition anta. Here we will write genotypes. Illi offsprings anta bariyona. So parents alli man condition homozygous iddaga, IA iddaga. ಅದೇ ರೀತಿ ವುಮೆನ್ ಕಂಡೀಷನ್ IB ಇದ್ದಾಗ, ರೈಟ್? ಸೊ ದ ಜೀನೋಟೈಪ್ ಏನಾಗತ್ತೆ ಆಫ್ ಸ್ಪ್ರಿಂಗ್ ಇಲ್ಲಿ IAB ಅಂದ್ರೆ ದ ಬ್ಲಡ್ ಗ್ರೂಪ್ ವಿಲ್ ಬಿ AB, ರೈಟ್? ಸೇಮ್ ವೇ ಇದು ಹೋಮೋಸೈಗಸ್ ಕಂಡೀಷನ್ ಆಯ್ತು. ಸೇಮ್ ವೇ ಅಗೇನ್ ಅದೇ ಟೇಬಲ್ ಅಲ್ಲಿ ಕಂಟಿನ್ಯೂ ಮಾಡ್ಬೇಕು ನಾವು. ಸೊ ಹಿಯರ್ ಮ್ಯಾನ್ ಇದ್ರೆ heterozygous condition ಅಲ್ಲಿ ಇದ್ದಾಗ IAI, we have to write heterozygous there. IBI ಇದ್ದಾಗ ಸೊ ಏನಾಗತ್ತೆ ಇಲ್ಲಿ IAI crossed with IBI. So IA is a gamete, I is a gamete, IB is a gamete, I is a gamete. IAIB AB, IAI A, IBI B, right? Then I into IO. So AB ABO. So ಇಲ್ಲಿ ಯಾವ ಫಿನೋಟೈಪ್ ಸಿಗತ್ತೆ ನಮಗೆ? ABO AB ಯಾವಾಗ? when both are heterozygous in condition. So instead of writing it in a points, if we draw the table like this and if we write alva, it is very easy and time ತುಂಬಾ ಕಡಿಮೆ ಕನ್ಸ್ಯೂಮ್ ಮಾಡುತ್ತೆ and presentation ಚೆನ್ನಾಗಿ ಅನ್ಸುತ್ತೆ. Okay, let us proceed to the next question. In a snapdragon, a cross between varieties, snapdragon plant ಇದೆ, ಅದರ ವೆರೈಟೀಸ್ ಕ್ರಾಸ್ ಅಂದ್ರೆ ಒಂದು ರೆಡ್ ಫ್ಲವರ್ ಇದೆ, ಇನ್ನೊಂದು ವೈಟ್ ಫ್ಲವರ್ ಇದೆ. ಅವೆರಡನ್ನ ಕ್ರಾಸ್ ಮಾಡಿದಾಗ ಇಟ್ ಪ್ರೊಡ್ಯೂಸಸ್ ಆಲ್ ಪಿಂಕ್ ಫ್ಲವರ್ಸ್ ಇನ್ ದ F1 progeny. Explain how it is possible, how it is a case of incomplete dominance. ಅದು ಇನ್ಕಂಪ್ಲೀಟ್ ಡಾಮಿನೆನ್ಸ್ ಹೇಗಾಗತ್ತೆ, ಯಾಕೆ ಬ್ಲೆಂಡಿಂಗ್ ಆಗೋದಿಲ್ಲ ಅಂತ ಕೇಳಿದ್ದಾರೆ. ಬ್ಲೆಂಡಿಂಗ್ ಅಂದ್ರೆ ಮಿಕ್ಸಿಂಗ್ ಅಪ್ ಆಗೋದಿಲ್ಲ ಅಂತ. Can anyone read this? Yes. Deepak, can you read this?
Yes, ma'am.
Yeah.
In incomplete dominance, the genes of an allelomorphic pair are not expressed as dominant and recessive, but express themselves partially when present together in a hybrid and is an intermediate between the two genes. As a result, an intermediate character is obtained. Example, two types of flower occur in Mirabilis jalapa, 4 o'clock plant, and Antirrhinum major snapdragon dog flower. The red flower color is due to gene RR, white flower color is due to gene small r, but pink flower color appears in case of genotype big R and small r.
Okay, thank you, Deepak. So here we have to write it in a point wise. I forgot to make it as a point. In incomplete dominance, the genes of allelomorphic pair, allelomorphic andre alleles iruva pair galenive, that is red and white, okay, are not expressed as dominant. Illi ಡಾಮಿನೆಂಟ್ ಮತ್ತೆ ರಿಸೆಸಿವ್ ಅನ್ನೋ ಆ ರೀತಿ ಎಕ್ಸ್ಪ್ರೆಷನ್ ಕೊಡೋದಿಲ್ಲ, ಬಟ್ ದೇ ವಿಲ್ ಎಕ್ಸ್ಪ್ರೆಸ್ themselves partial here in this. Ili, it is not a dominant. It will also express partially. It will also express partially. So both together will give a different phenotype. ಎರಡರ ಮಧ್ಯೆ ಇರೋ ಫಿನೋಟೈಪ್ ಏನಾಗತ್ತೆ, ಎಕ್ಸ್ಪ್ರೆಸ್ ಆಗತ್ತೆ. That is why here it is not a blending, it is a incomplete dominance. So this kind of incomplete dominance we can see in Mirabilis jalapa as well as Antirrhinum majus. The red flower color is due to gene RR, white flower color is due to gene RR, but pink flower color appears in case of genotype R and R, where red color is expressed partially and white color is expressed partially. Okay, so like this the thing happens. Fine. Next question. Okay. Mention the, now we completed 2 marks question, guys. Now we are starting with 5 marks question. Okay. Mention the chromosomal disorders that are due to trisomy represent their karyotype and 2 symptoms of each. Karyotype ಮತ್ತೆ symptoms ಅನ್ನ ನಾವು mention ಮಾಡಬೇಕು. So please write down this question, don't forget. So I will let you know how to write this kind of questions. Mention the chromosomal disorders that are due to trisomy, represent their karyotype and 2 symptoms each. Okay. Let us proceed. ಇಲ್ಲಿ ನೋಡಿ. Let me show you like this. Here we have to draw table like this, guys. ಯಾವ ರೀತಿ, ಫಸ್ಟ್ 3 genetic disorders ಇದೆ. ಎದ್ರ ಬಗ್ಗೆ, ಯಾವುದರ ಆಧಾರದ ಮೇಲೆ, ಟ್ರೈಸೋಮಿ. Based on trisomy, we have 3 genetic disorder. The first one is Down's syndrome. This Down's syndrome cause effect. It causes, it is caused by the defect. ಯಾವುದರಲ್ಲಿ ಡಿಫೆಕ್ಟ್? ಆಟೋಸೋಮ್ಸ್ ಗಳಲ್ಲಿ ಡಿಫೆಕ್ಟ್ ಆಗತ್ತೆ. It is trisomy of 21st chromosome. 21st chromosome ಅಲ್ಲಿ ಒಂದು ಎಕ್ಸ್ಟ್ರಾ ಕ್ರೋಮೋಸೋಮ್ ಬರುತ್ತೆ. So the total number of chromosome will be 47 and you have to write the symptoms. Next one, Patau syndrome. So Patau syndrome is also caused due to the trisomy, but ಇಲ್ಲಿ explain trisomy. Trisomy ಅಂತ ಅಂದ್ರೆ this is one chromosome, this is one chromosome. ಇದರ ಜೊತೆ ಇನ್ನೊಂದು ಕ್ರೋಮೋಸೋಮ್ ಇತ್ತಂದ್ರೆ it is called as trisomy. Okay, so here on trisomy of 13th chromosome, so ನಮ್ಮಲ್ಲಿ 13th chromosome ಅಲ್ಲಿ ಒಂದು ಎಕ್ಸ್ಟ್ರಾ ಕ್ರೋಮೋಸೋಮ್ ಬಂದಿರುತ್ತೆ. So due to that we will get a syndrome named Patau syndrome. Okay, and these are the symptoms of it: difficulty in gaining weight, extra finger or the toes, ears forming low on the head, growth abnormalities, low muscle tone, ತುಂಬಾ ಚಿಕ್ಕದಾಗಿರುತ್ತೆ ವಾಯ್ಸು. Next we have Edwards syndrome ಅಂತ ಬರುತ್ತೆ. It is also a defect in an autosome. It is caused due to trisomy of 18th chromosome. 18ನೇ ಕ್ರೋಮೋಸೋಮ್ ಅಲ್ಲಿ ಒಂದು ಎಕ್ಸ್ಟ್ರಾ ಕ್ರೋಮೋಸೋಮ್ ಇರುತ್ತೆ. So what are the symptoms? Low birth weight, small head and jaw, unusual looking face. So ನಾನು ಯಾಕೆ ಈ ಒಂದು ಟೇಬಲ್ ಅನ್ನ ನಿಮಗೆ ತೋರಿಸಿದೆ ಅಂತಂದ್ರೆ, ಲೈಕ್ ದಿಸ್ ಯು ಹ್ಯಾವ್ ಟು ಪ್ರೆಸೆಂಟ್ ದ ಆನ್ಸರ್. ಸುಮ್ನೆ ಡೌನ್ ಸಿಂಡ್ರೋಮ್ ಅಂತ ಬರೋದು, ಅದರ ಜೀನೋ ಟೈಪ್ ಬರೋದು, ಅದರದೆಲ್ಲ ಸಿಂಪ್ಟಮ್ಸ್ ಬರೋದು, ನೆಕ್ಸ್ಟ್ ಅಗೇನ್ ಪಟಾವ್ ಸಿಂಡ್ರೋಮ್ ಬರಿತಾ ಹೋಗೋ ಬದಲು ಇಫ್ ಯು ರಿಪ್ರೆಸೆಂಟ್ ಲೈಕ್ ದಿಸ್ ಇಟ್ ವಿಲ್ ಬಿ ಗುಡ್. ದ ಪ್ರೆಸೆಂಟೇಶನ್ ವಿಲ್ ಬಿ ಆಲ್ಸೋ ಗುಡ್. ಮತ್ತೆ ನಿಮ್ಗೆ ಓದೋವಾಗ್ಲೂ ತುಂಬಾ ಬೇಗ ನೆನಪಲ್ಲಿ ಉಳಿಯತ್ತೆ. ಇದು ತುಂಬಾ ಈಸಿಯಾಗೂ ಬರಿಬಹುದು. ಡಿಡ್ ಯು ಗೆಟ್ ದಿಸ್ ಗೈಸ್? ಅರ್ಥ ಆಯ್ತಾ? Deepak, whether your doubt got resolved? Klinefelter syndrome ಅಲ್ಲಿ ಏನಾಗತ್ತೆ, 1XY ಕ್ರೋಮೋಸೋಮ್. ಸೊ ಇಲ್ಲಿ XXY ಬರುತ್ತೆ ಅಲ್ವಾ Klinefelter syndrome ಅಲ್ಲಿ. And it is related to allosome. It is related to sex chromosome. ಕೆಳಗಡೆ ಇದೆ ಆ ಟೇಬಲ್ ಅಲ್ಲಿ. ಬಟ್ ನೌ ಐ ಆಮ್ ಡಿಸ್ಕಸಿಂಗ್ ಓನ್ಲಿ ರಿಲೇಟೆಡ್ ಟು ಆಟೋಸೋಮ್ಸ್. ಆಟೋಸೋಮ್ಸ್ಗೆ ಸಂಬಂಧವಿರುವ ಜೆನೆಟಿಕ್ ಡಿಸಾರ್ಡರ್ಸ್ ಅನ್ನ ಮಾತ್ರ ನಾನು ಡಿಸ್ಕಸ್ ಮಾಡ್ತಿದ್ದೇನೆ. ಡಿಡ್ ಯು ಗೆಟ್ ಇಟ್ ದೀಪಕ್? Okay, fine. Okay. So we have many more 5 marks question which we will be discussing in the next sessions. Okay, any doubt? Any doubt? Yes, YouTube students, any doubt? Okay. Okay, then let me end up the session. Have a nice day. Take care. Bye-bye.